What do you want? We have a little helper. What you doing? She is very, very cute. Okay. All right. Before I get distracted, you're going to be a real problem sitting there. You know that, don't you? Hey, shift your butt. Can't I can't erase part of the whiteboard now. I guess I am. All right. So let's get on to chi-square goodness of fit. We're going back over there. Good. Sorry, a little bit late because on the way back to the couch, discovered a pee on the floor. So that had to get cleaned up. Anyway, so kind of getting back to this, this is really a test to determine whether or not what you predict or expect is happening is actually happening when you do the experiment and look at the actual numbers, the observation. And so first, before we do anything, and this is something you have to keep very much in mind here, right? Because statistical tests test different things. So a statistical test that is actually looking to determine if there's a difference between two groups is going to give you a different answer to this kind of test, which is test and see if what you expect or what you observe matches what you expect, right? That's something that you got to bear in mind, because otherwise it can be kind of confusing, particularly as the chi-square test works a little bit differently. So first things first, we've got to talk about the term statistically significant, right? And so if you're a bit of a science pedant like me, you'll start noticing, like in literature and the news, whatever, when people say things are statistically different, what they normally mean is that they look different, right? What they should be meaning is that they've done the statistical analysis to determine if they are statistically different, right? And so this has a very particular meaning, right? And what it's saying is that you've done an appropriate test and you've determined that the difference that you observe, right? Because it'll almost always be differences between either what you expect and what you observe in this kind of test, or between two different groups, say your experimental and your control, right? What you really care about is that difference due to chance? If it is due to chance, what that's saying is that you repeat this again, bloody ears come back again. If it is due to chance, then if you do this again, then the second time you do it, that difference may not be there. Or it may be in the other direction, right? Instead of getting more than what you expect, you may get fewer than what you expect because it's just due to chance. Essentially, it is the noise in the system, right? Or a consequence of maybe you're not doing a big enough sample size to really tell if something is different or not. That's a kind of the other side. Having a puppy at your feet is very distracting. Now, if it is statistically significant, or if there is a statistically significant difference, what that's saying is that this difference that you're observing, or that comes out of this statistical test, is not due to chance, or is unlikely to be due to chance. Because if the kind of the follow on from that is if it's not due to chance, then something must be causing it. Right? That's kind of the key point to get across. If chance is not responsible for this difference, then something else is. And that something might be something you're interested in, say adding a particular type of fertilizer to a crop and you get better yields, or giving a patient a particular drug and getting less disease or something like that, right? In which case you're the responsible party, right, when it comes to the cause of that difference. Or in the case of a goodness of fit test, it may be that your predictions were incorrect. That's essentially what it's telling you. So if your predictions are correct, even if your observations differ a little bit, then that difference is likely to be due to chance, i.e. it's not statistically significant. If there's a large difference, then after a certain point, your statistical test will say, hang on, there's something going on here, right? Because what you expected doesn't match what you observe, or vice versa, really. And so therefore, something is wrong, right? And typically that means that your expectations were wrong. So your hypothesis is wrong, right? Therefore, something else is going on, which you're not really kind of capturing with that hypothesis. And so we use a threshold. It's kind of like a line in the sand, essentially, somewhat arbitrarily chosen. And that significance limit, right, or really probability, is the probability of getting that result by chance is 1/20th or less, right? So really, that's the p-value. Hang on, it should be that. So what that's telling you is that the probability of getting this difference by chance is less than 1 in 20. And obviously a smaller number, so let's say if you had p-value equals 0.001, that's telling you that the probability, I hate writing those words out. That one, that spelling mistake will have to stay. And that's 1 in 1,000. So the smaller the p-value, the lower the likelihood that that difference that you're observing is by chance. And so if it's not by chance, it's got to be by something else. And that something else is typically the thing that you're interested in. All right, so that's really what you're looking at. Jordan, what's up?
Sorry, sir. How did you word the p-value? I was trying to write that down, but I didn't finish in time.
That's okay. The p-value is the probability of getting this difference or the difference you observe by chance.
Okay, gotcha. Thank you.
Yeah. And so the smaller the p-value, the lower the probability that that difference occurred by chance alone. So essentially something else is causing that difference. And actually it's very cute. And so with the chi-squared test, what we're interested in, what we care about, is how well do our observed data fit our expectations? And so if our expectations are correct, there will be a small difference between our observed data and our expected data. And so our p-value, or the probability of that getting that difference by chance, will be pretty high. Now, if you completely screwed up your expectations and you got the totally wrong hypothesis, there'll be a large difference between what you observe and what you expect. And so that difference will be, the probability of getting that difference by chance will be very, very small. And so that's what we're testing with the chi-squared test. And so there's a bunch of steps, right? So this is really the kind of formula, right? So chi-squared, that's what this thing is, right? This funky E, which we've come across in lab when we're looking at mean and standard deviation, that's the sum of. So for every different condition, let's say, you're looking at amount of round peas versus wrinkled peas, for example. Right. For each condition, you're going to have your observed data, right? So how many round peas did you get? And then your expected number of round peas. So based on Mendelian principles and probability, how many round peas would you expect from that number of peas in total? And then your chi-squared test is this calculation, which is observed minus expected squared. And we, like with variance, we square it because there's going to be some which are larger and some which are smaller than that. So that kind of gets rid of the negative values. And then you divide that by expected, right? Because this accounts for the scale of what you're looking at. Jordan.
Yes, sir. I'm just trying to make sure I understand the equation. The sum is just for the numerator part of this equation, correct? Expected is over everything.
Give me a second. I'll write it out in the chat. It'll be a little bit. Actually, I'll write it out on the board. That'll be easier. So if you have two different outcomes, I guess, conditions, phenotypes, whatever you want to call it, your chi-square will be the observed minus the expected squared over the expected for outcome 1 plus the observed minus the expected squared. Oh man, my writing is terrible. Over expected for outcome 2, or phenotype if you want to use that instead.
So the sum would be in that original. The sum was everything following that formula or following the sum symbol, correct?
Correct. So the sum is basically saying that the original that equation up there is for however many different phenotypes or outcomes you have, you calculate the square of the difference basically over the expected for each of those, and then you add them all together.
Gotcha. Is there any chance this is on Excel as well? Or is this something we have to do individually?
Not in Excel, but if you want to teach yourself how to use R, it's a real piece of cake in that, supposedly.
What was the website, sir? Or what was the application?
R is just the letter R.
R.
It's the pirate's favorite statistical software, if pirates did statistics.
And could I find it online, or do I have to download, or is it like a website, or do we have to download it?
It's an actual program. This is something that when you do statistics and biology with Dr. Page, you will learn how to use R. So right now, I would worry about the crab sticks, the actual mechanics of what we're doing. And if you have the time and interest, you can do this in R. And if not, I won't be giving you anything so complicated that you wouldn't be able to do it, one of these in like 5 minutes or so.
Okay, gotcha. Awesome. Thank you.
Yeah, but definitely as you get more kind of competent at these kinds of biological statistics, you're going to want to use software or websites to do these kinds of tests. Without a doubt. What did Dipper put in there? Huh. Yeah, I mean, R is actually pretty cool. I know exactly just what I need to, how to do what I need to do. I actually have a list of instructions written down so I don't forget for working out specific things, which Dr. Page gave me, but I really don't know how to use it any more than that. But it's a very powerful piece of software. So this might not make a whole bunch of sense to you right now. But this is a fairly straightforward way of doing a chi-squared test. So first, you got to work out your expected. Right. And this is based on your expectations, right? Your predictions that come from your hypothesis and the total sum of everything that you observe. Right. So if we have, let's say we're doing let's get this share off so it's a little bit bigger. Doing a monohybrid cross, we would expect 3/4 dominant, 1/4 recessive, right? Based in the F2. Based on our knowledge of Mendelian inheritance and how a monohybrid cross is structured, right? So you have to start with your hypothesis and the predictions that that hypothesis generates. Does that make sense?
So like I'm trying to think of it as just like flipping a coin. So if you were to flip a coin 50 times and you know that there's a probability that it's going to hit heads or tails, your expected would be like 25 heads and 25 tails. Is that what you, am I thinking of that
yes,
right?
Yes, you are. You're jumping ahead just a little bit.
Oh, okay.
So your hypothesis would be that you would get an equal number of heads and tails, right? So actually, let's use your example. So Marissa, this isn't a given. This is based on your hypothesis and your knowledge of the situation, like how things work. So yeah, good example, Robert. Let's use that. So your expectation will be half heads, half tails, right? So to work out your expected, actually, let's put values up here. That's not going to hurt. And move my laptop a little bit. So your expected values, if you flip a coin 100 times, so expected heads equals 100 times a half equals 50. Expected tails is the same, 100 times a half equals 50. Right, so that's the very first thing you have to do in a chi-squared test. What's your hypothesis? Well, my hypothesis is there's nothing affecting whether you get heads or tails in a coin flip. So I would predict that I would get half my total would be heads and half would be tails. So if you do 100 times, then you times your total by your probability of getting that given outcome. Now, if we were doing a monohybrid cross with peas and you scored 3,520 peas, then that would be your total times the probability of getting a particular outcome. Right, so if you're looking at a dominant recessive trait, it'll be 3,520 times 3/4. That will give you the expected dominant. So this is all, apart from your actual total, this is all theoretical right now. So this is based on your hypothesis. This is what you expect to get, not necessarily what you do get. So, everybody with me so far? Did that make sense, Marissa?
Yes, that does make sense.
Okay,
Thank you.
cool. So I'm a big fan of like the slow and steady route to success when it comes to mathematics. It's like I usually have a 3-step plan for pretty much everything. So the second one is work out chi-squared value. Right? And so if we go back to that equation where chi-squared equals sum of observed minus expected squared over expected, right? And now let's say we got 49 heads and 51 tails, right? So we didn't get exactly what we expect, which should be 50-50. Now what we want to know is that difference between what we observed and what we expect, is that of any interest? Is it meaningful? That's what this is going to tell us. So the first one would be for heads, which will be, let's say 49 minus 50 squared over 50, because we expected 50 heads. We actually got 49, right? Oops. But we also have to do the other condition, right, which is tails. So that's going to be 51 minus 50 squared over 50. And that will be, oh, actually 1 over 50 is 0.02. So that'll be 0.02 plus 0.02. Is that right? Yeah, that's right. Equals 0.04. I already get how I got that number. Essentially, it's 1 squared, which is 1, over 50. 1 over 50 is 2 over 100, or 0.02. And then you get the same for this one too. Hopefully that calculation is right. I can do basic stuff in my head. However, that is only the chi-squared value. I really gotta get myself a new camera.
So, sir, are we gonna find the square root of the 0.04?
No, there's no more square rooting or anything. That's simply the value that we're going to use.
So that just represent, okay, I guess I was thinking chi-square, we're trying to find the x ultimately. Because it looks like we have a square root or a
No,
squared, yeah,
this is what we care about. We're not going to do anything to this number after this, but this number alone isn't going to give us the answer.
okay.
Okay, so the third step of our 3-point plan to chi-squared success to work out the p-value. And the reason for that is, sorry, Marissa, you get to a second, is that that difference is going to matter, whether it matters or not, or whether it's important or not, depends on how many different conditions you have. Right, because oh no, that's okay. Every different, let's say if you have 20 different conditions, right, each with a difference, then you're going to add all those together, you'll get a much bigger number, right? So we need to take into account how many different conditions we have. So one, need the chi-square value, which is 0.04, right, which we got from step 2. 2, we need the degrees, oh, if I can spell degrees, of freedom, called df. And that's to take into account how many different conditions we have. And df equals n minus 1. So in this case, we've got 2 different conditions, tails or heads. So it's 2 minus 1 equals 1. This piece of information, chi-squared value, and this piece of information, degrees of freedom, are what we need to work out the p-value. And then you use a chi-square table, because I can tell you what, I'm pretty sure that this isn't significant because it's a very small difference. But now we go back to actually, I'm going to show you let's see. So you can use one of two things. You can either use a chi-square value chart or a chi-square value table. Okay, so essentially there's 3 different bits of information for a chi-squared test. Chi-squared value, degrees of freedom, p-value. So you're going to use your chi-squared value and the degrees of freedom to work out the third value, which is the p-value. And so what you do is first you find your degrees of freedom, which in this case is 1, right? So you start off on this row. And now you see, where does my value fit? And so 0.04 is basically higher than the lowest it's smaller than the smallest value that we have on here. Right, so come on, chat, where are you at? So our chi-squared value of 0.04 is smaller than that for p equals 0.5. It's the essentially what you have, the chi-square values are the ones in the middle of the chart rows. So each column represents a probability level. And so what you're looking for is, where does your chi-squared value fit in one of these columns? So is it larger or smaller than any of these numbers? And so our one, 0.04, is smaller than this number here in the first column. And so then we read up and we say, okay, that's a p-value of 0.5. Actually, it's smaller than that because it's, you know, even further kind of to the left. So what that tells us is that the likelihood of getting this result again by chance, right? So say you got a coin and you flipped it another 100 times, there's a 50% probability that you would get a different result from what you got this time. And it would be a small difference, again, just by chance. Now, let's say we did this, and again, keeping with the coin flip analogy, right? Let's say we expected 50-50 heads, tails. That would be statistically insignificant or not statistically significant. Tails. And we observed, let's say, 25 heads and 75 tails. So now we do a chi-square test. And this is just kind of illustrate kind of the purpose, I guess, of this. And so our chi-squared value would be, actually, what numbers am I going to put in? Our observed for heads is 25. What's our expected? 50, right? So it's 25 minus 50. We square that and divide it by 50. Plus, then we do the tails. 75 again, minus 50. Squared divided by 50. So,
Is it 50? I'm sorry, is it 50? Because it can be either or.
correct. So the 50 is because of our expectations. So, you know, given that there's an equal probability of getting a heads or a tails, then our probability of getting either of those is a half, right?
Oh, right. That's still going. Okay, so I'm sorry. That's from coming back from step one. We're still using those same numbers. Okay, thank you.
Yeah. So in step one, you take your expected probability, which is a half for heads, and you times that by the total number of observed events or counts or whatever. That's the very first thing. So however many heads and however many tails you get, you add those together, and that's the number that you times by the probability for getting one of those. That's actually a very key point. It's a good question. It's a very key point in doing this, one that students sometimes don't get. You have to take the total number of observations and times that by the probability of the one that you're interested in, your expected probability. That will give your expected number for that particular outcome. Does that make sense?
Yes, and then so you'll be giving us the observed so we know how to do, right? The observed will be given by you. Okay.
Correct. Actually, typically what I'll do is I'll give you the let's say I'll give you the cross that we're doing, and I'll give you the observed data, and then I'll ask you, is there a significant difference between what you expected and what you observed? That's a pretty standard question for this kind of stuff. So you'll actually have to work out the chi-squared value and degrees of freedom, and then use one of these tables to be able to figure out what the p-value is.
Thank you.
No worries. So if we follow this through, then we're going to have 25 squared divided by 50 plus 25 squared divided by 50 equals, what is 25 squared? That's what it is, divided by 50. So that is 12.5 plus 12.5. You're getting how I got those numbers. So 25 squared is 625 divided by 50 gives you 12.5 equals 25. What are the degrees of freedom in this test? Correct. Thank you, Rose. So now we go back to this chart. We're still on the first row, degrees of freedom 1. And now we look down here and we see, whoa, 25 is larger than the largest number. So that tells us, hang on, let's write this up. Right? And so from that, we can say that the p-value is smaller than 0.001.
And sir, for getting the DF, it's n minus 1. Where did we get the n from?
The n is essentially the number of calculations you did. So we have heads and tails. So we have 2 different calculations, and it's 2 minus 1. So that's what the n is really. It's the number of different phenotypes or outcomes.
All the sums, right? Like at the end of the sum. Okay. All right. Thank you.
So for example, if you're doing this with a dihybrid cross in the F2 generation, you'd have 4 different phenotype combinations. So in that case, your degrees of freedom will be 4 minus 1, which will be 3. So yes, Rose, this would be highly significant. That's what I was about to type next. So that is, any difference is highly unlikely to be due to chance. And so you saw a very large difference between what you expected and what you actually observed. So something is going on, right? What that something is, this test does not tell you, right? You need to go and do some investigation. Now, it might be someone's playing with a Dicky coin, right? And so the coin is weighted. That's probably actually about all I can think of when it comes to a coin toss. They're fairly straightforward things, right? Or, you know, for some of the flips, the person doing the flipping flipped in one with 2 tails rather than the heads and the tails or something, right? Something is going on that is producing this different that's not due to chance. So that's really what the chi-squared is all about. It's about telling us is the difference we see, is that difference due to chance or is it due to something else? And that something else is something which means our expectations do not match our observations. And in the, so for example, if we were doing a chi-squared for a dihybrid cross, we had 4 different phenotype combinations. Now our degrees of freedom will be 3. And now we would look along here for where our chi-squared value fits in. So for example, if we got a chi-squared value of 8, that would be larger than this one and lower than this one. So what we could say is that our p-value is lower than 0.05. Can't actually tell exactly what it is. You could use this chart if you wished, which actually kind of makes sense if you kind of think through it. It's just not as intuitive. But we would say that that was a significant difference because the p-value was lower than 0.05. Anybody got any questions? Okay, so what I would suggest doing is work through this problem and get some practice. Because this is really straightforward once you've done it a few times. Just follow those, those 3 different steps. Work out your expected values first, work out your chi-squared value and degrees of freedom, and then work out your p-value from that. Cool. Anybody got any questions?
And Professor, that was actually one of the questions I had wanted to ask you. So I was looking at this previously and I was a bit confused on how you got the 0.75 and 0.25.
Okay.
And how it correlated to the 99, or I'm sorry. Never mind. It was just that part of where you got the 0.75 and 25.
Okay. So these are our probabilities based on our hypothesis, essentially, right? And so in a monohybrid cross, we always know exactly what happens at each step, right? And so in our F2 generation, we'll always expect 3/4 of our progeny to show the dominant or wild type phenotype, and 1/4 of our progeny to show the mutant or recessive phenotype. So when you turn those expectations into decimals, we have 0.75 and 0.25. And so for any number, right, we could have scored 1,000 progeny, or we could have scored 15, which probably wouldn't have been a good idea. But anyway, whatever our total is, we times that by 0.75, and that will give us our expected wild type or dominant progeny. So the one mistake that students quite often make is that they don't use the total, right? You have to add these 2 different groups together to get your total, and then you work out your expected based on the total, not the individual numbers, but the total numbers. So you have to add those together first.
And then our resultant from the chi-squared, that's going to be our DF, degree of freedom.
So our chi-squared value is essentially the sum of the differences between our expected and our observed. We then need to put that into kind of scale, essentially, perspective maybe, by using the degrees of freedom. And that's the number of different conditions minus 1. So, sorry, just watching my daughter struggle through the door with a bunch of packages. So if we have 2 different conditions, it'll be degree of freedom will be 1. If we have 30 different conditions, the degrees of freedom will be 29 because it'll be n minus 1. So you need those first to do the chi-squared. You need your expected values, which are what these are here. Oh, and your observed, obviously, as well. Then you work out your chi-square value in your degrees of freedom. And with that, you can work out your p-value. So if you went through this problem, you get chi-squared value of 3. You then need to work out your degrees of freedom. And then you need to take that to this table or the chart. This is in the exams and quizzes section on Blackboard, because you'll be using it in the exam. And you look here and see, where does the value 3 lie on this row, right? So you look at that and it says, well, 3 is between a p-value of 0.1 and a p-value of 0.05. And so what that tells us is, even though it's a fairly large number, more or less, it's lower than that for the p-value of 0.05. So p-value is between these 2 numbers. So it's higher than 0.05. You don't actually care whether it's lower than 0.1 because it's still not significant. All you care about is that chi-squared value for that degree of freedom higher or lower than the number for p-value of 0.05. Does that make sense?
Yeah, just a little bit.
Okay. So again, work through it. These are things that which you learn by doing, really. Like, just listen to me chunter on. It's like, may make some sense, it may not. But this is really what you have to do by going through these and practicing them. And so find some examples. You can even just make them up. That's what I do typically whenever I teach them. Like, for example, the heads and tails thing. Never used that before. Just make up some values and work through a chi-squared test using those 3 different steps. Work out your expected values based on your expected outcomes. Love, I am teaching. My youngest thought it'd be a good idea to introduce one of our cats to the puppy. It didn't go very well for the cat, or actually for my daughter. So back to the program. Step 1, work out the expected values based on your expectations, right, which come from your hypothesis. Step 2, work out the chi-squared value by doing that sum, observed minus expected squared over expected, and then add them together. And then work out your degrees of freedom and use those 2 to work out your p-value. And again, what you care about is your chi-squared value higher or lower than the one in this column, this 0.05 column. If the chi-squared value goes to the right, is higher than that number, then it's significant. If it goes to the left, or it's lower than that number, then it is not significant. But really practice these, because that's the way that you get good at this stuff and you get quick at it, right? So that you're not spending a lot of time trying to figure this out, and instead of actually answering other questions in the exam. Any other questions? Cool. If you do have any, bring them to class on Thursday and we'll go through stuff, right? We can do some more, as long as we have enough time, we can do some more problems. That's what I try and do with all of this stuff, because you really learn by going through these things and doing them over again in different scenarios and getting a feel for it. So try practicing those yourself as well. Oh, I wish I could just lie on the couch and sleep like my puppy is. Anyway, so now moving on to mitosis. That's all right. So really mitosis is kind of, in a way we teach this first because we get to cover the basics of cell division. So it has value in learning it for that alone, right? So we get to understand more complex stuff. Rose, what's up?
I had a question I put in chat as far as for the chi-squared.
Oh yes, I'm sorry. My chat had disappeared when I changed PowerPoints.
That's okay.
So how would you use the second chart? Let's go back.
Sorry, I didn't want to move on until.
No, it's okay. I'd much rather spend time getting stuff right than skip on and assume you didn't. So here, essentially, rather than degrees of freedom being a series of rows, now they're lines on the chart. So essentially you're using that line. It's almost like a line of best fit, right? You're using the chi-squared value to calculate the p-value. So if we use that prior example of chi-squared value of 3 and a degrees of freedom of 1, you go down here, you can see where I'm pointing to. Find 3 on the x-axis, and you go up till you hit the line for 1 degree of freedom. And then you go across to the y-axis and you read off the p-value. So that will give you a p-value of 0.08. These are all stuff that's been calculated by other people, thankfully. You don't have to do it yourself. Now, if you wanted to use the other chi-squared value that we got, which was 0.04, that's probably over here, right at the end of the graph, right? So now you go up to the line, let's say we're halfway between these two. That's going to give you a p-value of 0.9. So there's a 90% chance that whatever difference you saw was just random noise occurred by chance. So that's how you use this chart there. The values are exactly the same as for the table, right? So the actual information in the chart isn't different. It's just a different way of using that information. So here you're essentially using the chi-squared value and the degrees of freedom to work out the p-value over here. On the chart, you're using the degrees of freedom and the p-value to work out, and the chi-squared value, sorry, to work out the p-value up here. The information is the same, it just different way of getting at it. So my advice is just use whichever makes most sense to you. That help? Let's put the chat up, see if I missed anything. Oh, dominant versus recessive, doesn't matter. So in the example here, you could just use the words wild type and mutant, or normal versus abnormal. But based on your expectations, where you expect 3/4 to be wild type or normal and 1/4 to be mutant or abnormal, that's how you work out your expected numbers. So the actual things that you're looking at, as long as you're consistent between, you know, what you expect and what you're observing, doesn't matter what these are. You're simply looking at the, give me a second, everybody. You're looking at the difference between what you observe and what you expect for that given outcome. Okay, who had questions? Jordan, you want to go next?
Sure. I just wanted to ask, is wild type one of those genetics terms that we should know and just be used to saying that or
yeah.
something?
It is. It's basically a fancy way of saying normal.
Gotcha. Wild type.
Yeah.
Is that just saying that this is what we typically see in the wild, I guess?
Correct. So if we're talking about a particular gene, this is what the version of that gene we see most commonly that produces a functional protein.
Okay, awesome.
So
Thanks.
it's kind of it's like the word normal, right? Determines on, that depends on what you call normal. Right, so you know, wild type in one population might be different from wild type in another, but at the end of the day, I don't know, non-shiny would be the Pokemon version of wild type, right? It's what you would think would be normal, most common. A mutant is obviously something that's different from that. But again, the terms really aren't super important, because really all you're doing is calculating the difference between observed and expected. So you could call these anything, really. Shiny or non-shiny, for example, non-shiny, shiny, if we're talking about Pokemon. Did that help, Rose?
Yes, that's where I was confused.
Yeah, so, and that's, I should apologize for that. This is just kind of genetic speak, which I'm used to using, and I forget that other people don't. Cool. Okay. Stroke some puppy ears while I talk about mitosis. So, one reason why we care about mitosis is that it's the way that we become who we are, essentially, right? After fertilization, we all began, believe it or not, it's kind of a pretty funky thing to consider. We all began as one single cell, a single-celled organism. We didn't stay that way for very long, because we then, all of us, without exception, underwent our first round of mitosis, which is cell division of somatic cells, right? And essentially, us as individuals, or you know, even little bit down there, who's keeping me company, we all have gone through hundreds, if not thousands, of rounds of cell division of mitosis and differentiation and specification and so on and so forth, to end up as complex multicellular organisms. Even simple organisms such as yeast also go through mitosis. But instead of getting large world-conquering complex yeast organisms, you just get more yeast cells, right? So it's basically the way in which you generate more of the same. That's kind of the key point about mitosis. You'll get 2 identical daughter cells from each round of mitosis. Now, that doesn't necessarily tell you whether or not what those daughter cells will become. That's a different kettle of fish. But you will get 2 identical daughters, right? And so the other reason why we care about this is that there are many checks and balances that make sure that this doesn't happen when it's not supposed to, right? And essentially, cancer or tumors are when mitosis begins again, essentially when development starts again in an inappropriate context, right? So that's why tumor cells are typically large clumps of undifferentiated, unspecified cells. Right, they may all be lung cells, but they're not like alveoli or capillaries or whatever. They're just large clumps of dividing cells because they're essentially restarting development, but without having all of the cues and the context and that, which would tell those cells what they're supposed to become, right? So mitosis is kind of like this, I wouldn't say a double-edged sword. That's kind of like over-egging it a little bit. But it has like these two sides to it. One is it's how we become who we are and how we replenish cells which get used up or degraded, like skin cells, intestinal cells, for example. But it's also something that if those checks and balances break down, you end up with tumorous cells, right, which are essentially dividing uncontrollably without any kind of context to tell them what they should become. So key to that, dog, sorry, she's wrapped herself around my power cord. Get that back. There we go. Key to all of this is the cell cycle, right? Oh, now she's gone off in a huff. So the cell cycle is really the whole picture of cell division. So mitosis, and this is actually where we get into a little bit of where we could be inconsistent or maybe confusing when we talk about what's going on. Mitosis, technically, correctly, like specifically, refers to the process of DNA or nuclear division during the cell cycle, right? This little bit here. However, we quite often refer to mitosis as being the generation of two identical daughter cells, whereas meiosis is the generation of 4 non-identical daughter cells, right? And so we quite often use mitosis and meiosis to refer to the whole process of cell division, not just the process of nuclear division during cell division. So that's kind of something where I try very hard whenever I ask you questions about this in the exam. I'll be very specific about the term that I'm using, but just be aware that there is that ambiguity in the use of these words, right? Mitosis can mean both nuclear division and also mitotic cell division, right? So just kind of get that out there. So the cell cycle, and this is a kind of applies to meiosis as well. Actually, it very much applies to meiosis. So really what we're talking about here are two main phases. We have the preparation phase, which is called interphase. And we have the M phase, which is really about divvying up DNA and cytoplasm. So M phase and cytokinesis, really. And so typically speaking, once cells have gone through cell division, there'll be two possible outcomes. One, that cell will divide again, right? That was kind of what you think about in terms of, you know, us going through development, lots and lots and lots of cell division. Or two, that cell may be done and it will no longer divide or go into a non-dividing phase. So a good example of that would be neurons. So all the neurons you're ever going to have in your whole life, you had when you were born. Actually, you end up with quite a few less neurons than that because you kill them off, right? So there's a neuronal die-off that occurs during early childhood, where you basically get rid of ones you don't need, right? So neurons are post-mitotic. Once they have been created through cell division, through mitosis, they will never divide again. That's why if you damage your brain or you damage, you know, other parts of your nervous system, it's very, very hard to repair them because those cells will not divide again to fill it. Contrast that with getting a cut or a scratch, like what I got on my finger after being stupid with an angle grinder. Your skin will just grow back together. Those cells will start dividing again and they'll fill that gap. So anyway, that's this G0 phase. Those cells are not dividing. They may re-enter cell division. Say if there's a particular cue, like if you have a stem cell in your skin or your intestine, or if one of those signals gets turned on accidentally, which then leads to cancer. And then you go through 3 different phases, right? And these are all about preparing the cell for the last step, which is the M phase. So you need to build all the machinery to copy everything that you need. You need to build a whole bunch of extra stuff like cell membranes and organelles and so on and so forth to divvy up during cytokinesis. And so the main point is to, at least in a G1 phase, you're going to generate everything you need for DNA replication. That's in S phase or synthesis phase. And in G2 phase, you're going to prepare the cell for mitosis by generating all the stuff needed for the M phase. Now, there are 3 really important parts of this cell cycle that I need to point out. And these are very interesting parts, and this is where errors occur or mistakes happen and things go wrong. And these are the checkpoints. So in every cell cycle or round of cell division, there are 3 different checkpoints, and they serve different purposes. And so the G1 to S checkpoint, so this occurs towards the end of the G1 phase or the gap phase. Its purpose is to check 2 things. 1, oh, I'm sorry, I missed give me a second, Adam, I'll get to your question. My chat box disappeared. First thing that the G, let's type this out actually. Checks for 2 things. 1, does the cell have enough energy to make it all the way through? Right, because this process takes a lot of energy. Copying all of your DNA and all of the proteins needed to do everything takes a lot of energy. And if you don't make it all the way through and you get stuck as a cell, then you die, right? Because you can't have a half divided cell hanging around. It will be told quite politely to kill itself through apoptosis. And the second thing that you're checking for is the DNA damaged? And similarly, you don't want a cell to divide that has damaged DNA, right? So those are the 2 things that G1 to S. Oh yeah, that's right. Yes, Adam, the key difference, other than number, is mitosis equals 2 identical daughter cells. Meiosis equals 4 non-identical, actually haploid daughter cells. But we'll get through that in more detail when we get to meiosis. on Thursday? Good question then. The second checkpoint is the G2 to M. So the G2 to M checkpoint coming after the S phase, its main job is to check to make sure all of the DNA has been replicated. Because again, you don't want to go into M phase where you divide up the DNA if you don't have 2 equal portions to give each daughter. That would be disastrous for many, many reasons. Pardon me. And then the final one, which we will just get through before we break for lunch, is the M phase checkpoint. Is the next one. And that's actually really, really funky. It's a really interesting, like the molecular biology and biochemistry behind it is pretty cool. And so what that does is it checks to make sure every chromosome pair, right? Because we have two chromosomes for each chromosome 1, 2, 3, whatever, because we're diploid, right? Every chromosome pair is aligned on the spindle. Basically, that's the metaphase plate. Or simpler terms, the center of the cell. And it'll wait for as long as it takes for that to happen. Bless you. And it's a very, very important checkpoint, because if that checkpoint fails, in other words, if it allows M phase to proceed without those chromosomes lined up all neatly, kind of down the center line of the cell, then one of those daughters will end up with 1 too few chromosomes, and the other one will end up with 1 too many. Now, this isn't super common in mitosis, but it is actually fairly common in meiosis. So breakdowns in the M phase checkpoint, those result in something that's called meiotic or mitotic, depending on the type of cell division, non-disjunction. And so that basically equals one daughter cell has 1 too many chromosomes. One has 1 too few. So in meiosis, that can lead to monosomy or trisomy. Does anybody know an example of trisomy? It's actually one of the most common birth defects in the US. You almost certainly know someone or have seen people that have this. Shush.
I'm just guessing. I don't know, cleft palate or something like that? I have no idea.
That is a good guess. That's actually due to developmental defects in signaling, where your brachial arches join together to create your mouth, your upper and lower jaw. Valeria has it. So Down syndrome is a classic case of the M phase checkpoint breaking down. So Down syndrome is trisomy 21, if I can spell that right. So you have 3 copies of chromosome 21. So one of the gametes that was involved in the fertilization of that individual had 1 copy of chromosome 21, the other one had 2. And so the resultant individual had 3 copies. Rose, autism is a very poorly understood process, and it's due to all bunch of potential things, both genetic and environmental. Actually, there are very few examples of monosomy and trisomy, because normally what happens if you have 1 too many or 1 too few chromosomes for a given chromosome is that you will not make it through development to being a functional individual. That will lead to miscarriage. Because it has such massive consequences. And you can actually get a sense of that by if you look at an individual with Down syndrome, not in a bad way, but you'll see that there are lots of different phenotypes that kind of come with that, right? They have a very particular craniofacial look, right? So you can always tell that someone has Down syndrome just by looking at them. They tend to be shorter in stature. They tend to have different kind of behaviors, right? They're very, very loving, kind of open individuals. So lots and lots of things are affected by this. And this is the most benign one that you can think of.
Sir,
Go on.
sorry, I didn't know if the hand raising thing. So you're saying Down syndrome is one of the few, I don't know how to word this right, few errors during mitosis that still lead to a viable
That's
organism.
correct.
Is there, just out of curiosity, is there any other common ones, just for my?
There's Klinefelters, which is that individual will have 2 X's and a Y. So they'll be genetically male, but partly physiologically female, and they're sterile. There's Edwards, where you are an X0. So you only have 1 X chromosome and no second sex chromosome. Those are genetically female, but are also sterile. There's one called Loup-Garou, which is a weird one. I don't actually know very much about it. That's actually a partial deletion of a chromosome, like part of it, not a complete lack of it.
Gotcha.
There aren't very many.
Yeah. So, are any of these things that we are going to delve into later in the semester, or
No,
is it just something to know in passing?
No, just kind of something to know in passing. Really, an illustration of how important that checkpoint is.
Gotcha.
Because if it fails, then almost certainly bad things will happen as a result. And one of the, so it's actually quite, I'll get to your question in a second, Rose. It's quite straightforward to think about the downsides of having only 1 chromosome, because essentially you don't have the second copy of that gene to potentially mask a recessive allele. And so that's the basis of sex-linked traits such as hemophilia or male pattern baldness or color blindness and things like that, right? You don't have the second chromosome to potentially have a dominant allele that will mask the recessive allele. So if you only have 1 chromosome 1, then if you have a whole bunch of recessive alleles for genes on the one that you do have, all of those will be expressed in terms of the phenotype, because you don't have the second copy. Now for trisomy, it's a little bit weirder, and basically that's a gene dosage issue. So you have, rather than 1 or 2 copies of your gene producing a protein, now you potentially have 3. And so that can just screw up a lot of other stuff like cell signaling pathways and stuff like that. Rose, what's your question?
I don't have a question, sorry.
Oh, that's okay. No worries. So it is a very, very interesting topic, and it's just kind of neat when you dive into it. We're not going to do a bunch of that right now because we're going to get onto the actual mechanics of mitosis and meiosis on Thursday. But it's just cool stuff to know, basically. All right, so run over a little bit. You're probably a little bit pie-eyed and brain blasted. So we'll meet up in half hour at 2:07, right? So a little later, just so we all have time to have our lunch and Robert can find another nice place to hang out at. So I'll see you in half an hour exactly. Okay, have a nice lunch.